This conference will now be recorded.
Hello students, I'm Dr. Divya from TeenEinstein. So now we are learning the sixth chapter, tissues, and this is the second part of this chapter. And in this chapter we are discussing about tissues. So our first topic is whether plants and animals, they are made up of same type of tissues. That is the first question when we are learning about tissues. So general concept about what is a tissue, that we have already covered in part 1. Then whether both the animals and plants both are composed of same type of tissue, that is the next question. Then plant tissues, what are plant tissues and what are animal tissues. These are the main topics which we are covering in this chapter. So here we said what are tissues, right? So in the previous chapter we have learned about cells and cell organelles, isn't it? So while learning about cell and cell organs, we learned that how a cell will work and what are the components of that cell and each component in the cell organelles. So those cell organelles, each and every cell organelle, how it will work or what is the function of each cell organelle, everything we have learned, right? So hope you remember that the previous chapter contents. And then we said there are unicellular organisms and multicellular organisms, right? The unicellular organism is they'll be having a single cell, and with that single cell it is performing all functions, right? But when we are coming to the case of multicellular organism, there we know that they have many cells, and here they have division of labor, right? Division of labor. What do you mean by division of labor? Their work. Multicellular organism is each and every cell cannot work to do the single functions. So all cells are not performing all the functions because it will be a complex body, right? Multicellular organisms will be having a complex body. So each and every cell is doing every work, means it's impossible. So what it will do? So it will become slow or it won't work in an efficient way. So what it will do? It will divide all the works. That body will divide all the works and different types of cells will be performing different functions. So each work, each and every work which is performed in the organism's body or in the body of a multicellular organism, there it is dividing the work. Okay, like respiration is done by one particular organ. So cells of that organ, they are coming under a group. In that itself, you can see different work is divided. Among in one organ itself, you can see cells are of different groups which are performing different functions, right? So here we have told about nerve cells, blood cells and all, which are doing their specific functions. So work is divided or division of labor. Labor means work that is divided. So there is a group of cells which is performing a particular function. So group of cells, that is called as tissues, and these tissues, some tissues will be assigned to do some work, right? And the cells of that tissue or a group of cells, that depends upon the function, right? Function, based on the function the structure is designed. Okay, then our next concept which we have learned, that is whether plants and animals They are made up of same type of tissues. See, for that we have learned like they don't have the same type. In a sense, we can say that they are not made up of same type of tissues, right? Because functionally and structurally, in structural way and in functional way, animals and plants have lot of difference, right? Animal body and plant body both are different. So the way how cells are functioning, that also will be different. The mode of functioning, that also will be different, right? Because one is absorbing what plants are doing. They are absorbing food and nutrients, or water and nutrients from the soil, and they have straight position, like they are fixed to the soil, and they have upright structure, right? So what they will do, they'll absorb nutrients and it should reach till the top of that plant body. So that needs stronger body structure, right? A supportive tissue, that is must. But compared to plants, animals have flexible body. Flexible, but this is somewhat upright, isn't it? Then another difference, growth pattern. Growth pattern, right? So growth pattern is also different. In plants, we can see some parts will be growing, or there the cell activity will be there throughout the life. But animals growth, up to a certain level it will grow, then it will attain maturity, then it will move to the reproductive stage, like that it is, right? But in plant body, it's not like that, right? So animal's body, that is more complex, the organ systems and the cells and tissues, then organ system, like that it is going. So that the structure is more complex in which one? Animal, right? Because more number of organs and organ systems are present, and they are working in a coordinated way to make the animal body functional. But here, compared to animal's body, it is less complex. Which one? Plant body is less complex. Okay, so we have learned that differences. So from that we can make out that animal cells and plant cells are not similar. Okay, then about we came to the topic of plant tissues, right? Generally we said animals and plants, they have differences in their tissues, right? So let's learn first plant tissues, then we'll go to the animal tissues. So plant tissues, for that we have done an activity, which is that onion root tip, we kept it for growing. In 2 jars we kept 2 onion bulbs, and we allowed them to grow, and then we are observing each, and after growing we are observing the growth of root, onion root, it is growing. So what will happen, we need to measure the length of root. First day, second day, third day, then fourth day we are selecting one jar among them, first jar or second jar, any one jar we are selecting, and we are cutting the root tip around 1 centimeter. Then we are observing whether the that cut portion that is growing, again we are measuring the length. So for which jar we have selected, that one will not show any further growth. Okay, so that we are making an observation from there. So from there we are getting some answers, one clear answer, like why the root, or which root is growing and which is not growing. Okay, and which one is having long root and which jar is not having long root. Okay, then here one jar we have selected, and we have done the removal of that root tip around 1 centimeter, whether that is continuing the growth continues in jar 2 or not. Okay, there after cutting the onion root tip in the second jar, we couldn't see any further growth. Okay, or we just like the experiment we have already conducted, we have already explained in the previous part. That's why I'm saying there we will not be able to see any further growth in the second jar. Why? Why would the tip stop growing in jar 2? That is the last question. Okay, here we are getting the complete answer for this activity. Why it is not growing? Because the growing region is located at the tip. So that part we have already removed. Around 1 centimeter we have removed. That's why that stopped growing. Right? So all questions, we got a clear idea about the growth pattern of that root. Right? So here, according to this experiment, we are coming to some conclusion like the growth of plants that is in specific regions. Growth is in some regions. It is in some regions, right? And it is pointed to some regions, and depending upon the region where it is present, this growing or dividing cells or the meristematic tissues are divided into three. See, plant tissues are divided into meristematic tissue and permanent tissue. And the meristematic tissue, based on the position where it is located, based on that, again, the meristematic tissue is divided into three types of meristematic tissues are there. Apical meristem, which is present at the tip, then intercalary meristem, which is present at the node, then lateral meristem, which are present laterally. Okay, so apical meristem is present at the growing tips of stem and root, and that helps to increase the length. Which one? Apical meristem, right? Apical meristem helps to increase the length. Okay, then lateral meristem, that increases lateral meristem. Here you can see the lateral meristem on side, and when it is dividing and cells, that time cells will get added to the sides, so that increases the girth of stem and root. Okay, then intercalary meristem, that you can see in some plants, which is located near the node. Okay, near the node. So cells of meristematic tissues, they are very active. Cells of meristematic tissues, generally we can say they are very active, and they have dense cytoplasm. They have dense cytoplasm. So about meristematic tissues, you can write they are in an active stage and they have dense cytoplasm, then prominent nuclei. Their nucleus is very prominent and they have dense cytoplasm. Then the cell wall is a rigid one, right? So that may interrupt cell division. That's why here cellulose wall, that is very thin, thin cellulose wall. So cellulose wall will be very thin and they'll be having a prominent nucleus, and they lack what? They lack vacuole. They lack vacuole. Okay. Why it is not having vacuole? Because vacuole, we mentioned, that is present in plant cell. In most of the plant cell, it will cover 50 to 90% of the cell, right? That was the point which we have learned in previous chapter about vacuole, right? But here in meristematic cell, why vacuole is absent? Here it is in the continuous stage of division, right? And what is the role of vacuole? I said in the previous chapter, vacuole, it's mainly meant for storage, right? Here continuously reactions are taking place, and it is the chemical composition of food synthesis is taking place inside that cell because it is in a meristematic stage or continuous division state, so that require more energy. So required more energy means that should be cell is in an active stage, that is continuously dividing, that require more energy, that so the cell should work, right? So there is no need of storage, there it is getting synthesized. Okay, so continuously energy is getting synthesized there, so no need of storage, so they lack vacuole. Clear? So here about meristematic cells, what you can say? Cells of this meristematic tissues. Meristematic tissue means group of cells which are in continuous stage of division, right? Tissues means group of cells. So meristematic means they are in a continuous stage of division. They are very active cells and they have dense cytoplasm. They have prominent nuclei, then thin cellulosic cell wall, and they lack vacuole. These are the main points which you need to mention while writing about cells of meristematic tissue. Clear? Next, so we have learned about meristematic tissues, right? Plant tissues are there of two types, meristematic tissues and permanent tissues. Meristematic tissues means they are in a stage of division. Continuously, they will be dividing, right? Next comes permanent tissue. So let's see what is permanent tissue. See here, meristematic tissue means that cells are continuously dividing, right? Those cells after division, again they will grow, they will attain maturity, then again it will go division. Then what will happen? Like that, continuous cells will get produced, right? Continuously that division is going on and the more number of cells are getting produced. So what is happening to the cells which is formed by the meristematic tissue? Cells formed by meristematic tissues. See, their continuous cell division is taking place, right? So what is happening to the cells which are formed by this meristematic tissue? See, they will take up some roles they will take up, that meristematic tissue. And finally, after doing that work so much, they will take up a role and they will lose the ability to divide. Okay, so they take up a specific function and they lose the ability to divide. So as a result of losing that ability to divide, they'll become permanent tissue. Then they'll become permanent tissue because they don't have the capacity to divide now. So they are becoming permanent tissue. So that process of taking up a permanent shape, then permanent size and a permanent function, that is called as differentiation. Okay, so what is differentiation? Differentiation of cell. See, this meristematic cells, they will continuously divide, right? So what will happen to these cells when this division is taking place? See, continuously dividing means numerous cells will be there. So after certain stage, after getting produced, these cells will take up some functions. These meristematic cells will take up some functions and they will lose the ability to divide. Then their dividing capacity they are losing. Then what will happen? They'll take up the function and they'll become a permanent tissue. Okay, so taking up of permanent shape, they will attain a permanent shape, they will a permanent size and permanent function also they will take up. Okay, that process, it is getting differentiated. That process is called as differentiation. Differentiation means they are attaining, they are getting separated and they are losing the meristematic activity. They are getting some permanent work, permanent size, permanent shape. Okay, they are getting differentiated from that meristematic tissues or meristematic cells and they are getting a permanent work, shape, size and all. Okay, so that cells are getting differentiated. That process is called as differentiation. Got it? Then see, here we are learning one activity, the second activity in 6th chapter. Okay, here with the help of teacher you can take a plant stem, then you can make into very thin slice. See, Suppose this is a plant stem, the young stem you need to select, okay, not the thick one. So if you're selecting that, from that you need to take thin slices. Thin slices you need to make, okay, or sections we'll call. In lab, if you're doing, we'll call it as sections, okay. Then we need to stain that slice with saffron, okay. We need to stain that thin section we are staining, okay. Then that stained one, so we'll mix the stain in a particular proportion. Then what will, after cutting that section, we will put in a glass slide. We'll mix the stain, and in that we'll keep this section, okay. Then that section, that cut section, we will keep on a glass slide, and we'll put a drop of glycerin, okay. So glycerin, why we are giving? In order to avoid the drying of that section. Then next we are covering with a cover slip, okay. Then we need to observe under a microscope. So first we are collecting a stem, which is a young stem, okay. Then we are taking thin sections from that. Then we are staining that. Next, what is that? Next, what we are doing after staining, we are keeping that thin section on a slide, and then giving one drop of glycerin to that in order to prevent the drying of that section. Then covering with the cover slip. While covering with cover slip, you should make sure that there is no air bubble inside it, okay. Then we can watch that under the microscope, okay. So when we are watching, we can observe. See, you will get a section like this. If you are taking the stem section, you can easily watch this kind of structures under microscope, okay. Various types of cells and their arrangement you can see, okay. This is the section of a stem, okay. So you can see how different types of cells are arranged in that, okay. So compare with this figure, keep this figure also with you, you can compare, okay. And here, this is the structure of dicot stem, okay. Dicots, plants are different types, dicots are there, monocots are there. So that and all you will learn in higher classes. So here the thing is, you keep this kind of diagram with you, then you can make out each and every structure. Now, just two questions they have given in this activity. First one is, are all the cells similar in structure? Here you can see different types of cells. See here, one bundle. See, this is the epidermis, the outer one, okay. Then phloem, xylem. See, this is one vascular bundle. If one section you are zooming, see, you can see here, this one section you are zooming, okay, one part. Then here you can see the outermost cuticle. Then comes the epidermis. Here you can see as a layers. So here comes the epidermis, a single layer of epidermis. Then comes collenchyma. Here you can see the collenchyma. Next, down to that two to three layers of collenchyma. Then you can see the parenchyma. Then phloem. See, here you can see phloem. Here you can see as a green line. So that one, when you're zooming, you'll get this kind of structure, phloem. Then xylem. Xylem, you can see towards inside, you can see xylem. Then this is the whole vascular bundle. Okay. Vascular bundle, you can see xylem and phloem. Okay. So that is the vascular bundle. Here you can see vascular bundle. So that is the section of a stem. You need to draw this and you need to mark like this. Okay. So here, how many types of cells you can see? You can see here, just notice this. Here one layer, single layer of structure, you can see cells, you can see. Then here Next to that, we have marked epidermis. That is one type of cell. Next comes the collenchyma, then the parenchyma. We'll learn about all those. So here you can see. And again, when we are coming to the vascular bundle portion, there itself you can see different types of cells, right? So different types of cells you can see in this section, isn't it? Here, now we are learning about different types of cell. While watching that section, which and all types of cells you can see in that. Okay. This is a stem section. If you're cutting a root tip and checking under a microscope also, you will see different types of structures. Okay. So in a root, it will be organized in some other ways because function of stem and root both are different, right? And here, next, when you're watching under microscope, you can see different types of cells, right? As a part of the permanent tissue, you can see different types of cells. So there are two types of permanent tissues. One is simple permanent tissue and another one is complex permanent tissue. Okay, so permanent tissues, that is of two types, simple permanent tissue and a complex permanent tissue. Okay, and the cells of these tissues, cells of tissues, tissue means group of cells, so the cells of that tissues, they have different functions. Okay, so here different types of permanent tissues are there. We said simple permanent tissue. First step is simple permanent tissues, simple and complex. These are the types of permanent tissue, simple permanent tissue and complex permanent tissue. In simple permanent tissue, you can see parenchyma, collenchyma and sclerenchyma. These are called as simple permanent tissue. Parenchyma, collenchyma and sclerenchyma. And here the complex permanent tissue, you can see phloem and xylem. Okay, xylem and phloem, they are coming under complex permanent tissues. Simple permanent tissue means those are simple tissues, but they are permanent tissues. Parenchyma, collenchyma, and sclerenchyma. Complex permanent tissues which are little more complex compared to the simple permanent tissue. They are phloem, xylem. Examples are phloem and xylem. Clear? So here you can see parenchyma, these cells. Then the collenchyma, you can see collenchyma, some more thick cells. Then sclerenchyma. Okay, so these are the simple permanent tissues. See here you can see the epidermis, right? Epidermis. So usually simple permanent tissues, you can see under the epidermis, few layers you can see. Few layers under epidermis. See, here you can see some layers under the epidermis cells that are simple permanent tissues. Okay. So here generally parenchyma, collenchyma and third one sclerenchyma. These are the main simple permanent tissues. So let's see one by one. Parenchyma. Parenchyma. This is the most common one. Most common simple permanent tissue. The first one, parenchyma, is the most common simple permanent tissue. Okay. Then they have, you can see here, they have very thin cell wall. Parenchyma cells have very thin cell wall. Okay, this is the longitudinal section and this is a transverse section. Okay, here you can see the cell wall is very thin. Cell walls are very thin, and uh, these are not specialized, unspecialized cells. Generally, they are unspecialized cells and they have very thin cell wall. And these are living cells. Which one? Parenchyma. They are living cells. Okay, we said these are permanent tissues, right? They have permanently attained structure, size, shape, and all. Okay, so these are the living cells. Okay, and usually they are loosely arranged. Loosely arranged. Here you can see a lot of gaps, and also they are loosely arranged, not arranged in a thick way. And they have large space between cells. Here you can see these are the spaces around. So space between cells you can see. So you can see loosely arranged, and they have space between cells, large space between cells, and these spaces are called as intercellular space. Inter means in between. Cellular means in between cells you can see the space. So they are intercellular. You can see intercellular space there. Okay. So in this group of cells or tissues, you can see intercellular space. Okay. And this is generally storing food. These cells generally that stores food. Which one? Parenchyma cells. Okay. So parenchyma cells, they are most common simple permanent tissues. And here, just remember this diagram. With the help of diagram, you need to write. Okay, so thin cell wall they have. It's not thick one. General thin cell wall you can see, and they are not specialized cells. Okay, unspecialized cells, and they are the living cells, and they are very loosely arranged. So it's loosely arranged because of that you can see space between the cells. Okay, which is called as intercellular space. And generally this parenchyma cells those stores food. Okay, and in some cases, see here in the longitudinal section we have marked chloroplast. So some parenchyma cells, see these are storing food, right? So some parenchyma cells in some kind situation they have chlorophyll and they'll perform photosynthesis. Chlorophyll, parenchyma cell with chlorophyll, they're called as chlorenchyma. Chlorenchyma cells with parenchyma cells with chlorophyll, they are called as chlorenchyma cells. And absolutely, when they have the chlorophyll content they can perform photosynthesis, and that kind of cells are called as chlorenchyma cells. And in aquatic plants means those plants which are present in water. So those plants they have parenchyma which are having larger cavities. So this intercellular space or the cavities will be more, or the larger cavities will be present, that is helping them to float. Okay, so that type of parenchyma cells in aquatic plants with large cavities, they are called as Aerenchyma. Aerenchyma cells, they are the parenchyma cells which are having large cavities, large air cavities which is present in that. So that is helping them to float. Such type of parenchyma cell is called Aerenchyma. Okay, so they are called as Aerenchyma cells. So this figure you need to learn. You need to mark vacuole, chloroplast, then the cell wall, cytoplasm, then here intercellular space. See the intercellular space you need to mark, and nucleus, then the cell wall, thin cell wall, everything you can mark and show. Okay, then another type of permanent tissue. in plants. That is collenchyma. Collenchyma, which is another type of permanent tissue, collenchyma. Okay, so this collenchyma is another type of permanent tissue, and that is giving flexibility. That is giving flexibility in plants. Okay, this is flexibility in plants, that is due to the help of another type of permanent tissue which is called as collenchyma. Okay, so this flexibility due to the presence of collenchyma, that is allowing the plant part to bend. This helps in the bending of plant part, right? So this is helping or allowing bending of different plant parts. So just think where we need that type of bending activity, like climbers, tendrils and all. In tendrils and climbers, in such type of plants or tendrils like plant part, there you need the help of this collenchyma to give the flexibility. Okay, so that it will climb without breaking. Without breaking, it will work. Okay, and that is providing, along with that, it is providing mechanical support also, right? It is giving mechanical support because it is working in climbers also, right? So we can find this type of tissues in leaf stalks below the epidermis. Okay, and these cells of this collenchyma tissues, they are also living cells. Living cells. Okay, and here you can see they are somewhat elongated, not like chlorenchyma. These are having elongated structure, and they are living, and they have some thickening in some area at the corners. Especially in the corners, they have thickening. You can see here this thickening, right? So they are thickened at the corners. At corners. So at the corner, they have thickenings. Okay, and here compared to parenchyma, they don't have that much intercellular space. Intercellular space is less. In which one? Intercellular space is very less. In which one? In collenchyma. Okay, so parenchyma we have learned, and this is collenchyma, which is giving flexibility to plant and plant parts. Okay, so flexibility in the sense that is helping to bend and to climb, and also in tendrils, climbers and all you can see, and without breaking, it is allowing that plant part to move, right? And climbers means that should climb on some other plant, so that in that way it is providing mechanical support also, right? And here the cells which are present in collenchyma tissues, they are living cells, and you can see in these type of cells, this is the diagram of collenchyma. This is the transverse section and this is the longitudinal section. So in this you can see, in the transverse section you can see the thickening at the corners, right? So in specific areas, especially in the corners, these cells are thickened. Okay, and here you can see the intercellular space, that is very less compared to parenchyma cells, right? So this is the simple permanent tissue, second type, collenchyma. Next, another, the third type, simple permanent tissue. Which is the third type of simple permanent tissue? Third type of simple permanent tissue, that is sclerenchyma. Here you can see sclerenchyma. Okay, sclerenchyma, this is another type of permanent tissue, and this tissue that makes the plant or plant part hard, and it makes stiff. This gives stiffness. So it is making the plant or plant part hard and stiff. right? We can tell like example coconut husk. Okay, so that is made up of sclerenchymatous tissue. And here we can see that coconut husk that is made up of sclerenchymatous tissue means here we can see the cells of these tissue. They are dead cells. Dead cells you can see in these tissues. Okay, so the cells of this tissue, they are dead. Okay, and here you can see they are long. This is the cross section. Okay, this longitudinal view. So they are long cells. Okay, and they are narrow also. Long and narrow cells. And here you can see the cell wall that is very thickened. Cell wall is thick, and this thickness is due to lignin deposition. Lignin deposition. So due to the deposition of lignin, these cell walls are very, very thick. And the cell walls are thick so that there is no internal space inside the cell. So cell walls are very thick. You can see thick cell wall. So there is no internal space due to the thickening of that cell wall. Okay, and this kind of tissue that is present in stem, where you can see in stem and around vascular bundle, then veins of leaves and hard covering of seeds and nuts. Hard covering of seeds and nuts. There also you can see sclerenchyma, and they are dead cells, and the cell wall thickening is more due to lignin deposition. Usually dead cells are there, and examples you can see coconut husk. There you can see sclerenchymatous cells which are dead. Okay. So which are the three types of simple permanent tissues? Parenchyma, collenchyma, and sclerenchyma. With diagram you need to learn, and the features or the characters of those cells of these simple permanent tissues also you need to learn. Clear? Next comes one activity. Okay, activity 6.3 of 9 chapter tissues. In that one activity is there. See, they're asking you to take a freshly plucked leaf of royo. Royo fresh leaf. Okay, then stretch that leaf. See, if you're stretching that leaf up to a limit, it will stretch, then what will happen? It will break. Right? If you're applying more pressure, it will break. Okay. Then after breaking, keep the stretched, keep it in that stretched way so that some skin, like if you're stretching that, some skin will peel off from that part and it will project out from the cut. Right? Skin peel will project. See, generally, that leaf, if you are stretching that, some peeled off skin will come and that will project. That will project out. Right? Then again, like how we did that section, stem section, like that, that peeled skin we need to take. That we are taking, then we are staining in saffronin, and under a microscope we are watching on a glass slide we are keeping it, then watching under microscope or observing under microscope. See, then you can observe this kind of cells. Okay, when you're observing the outermost layer of cells, you can see which are called as epidermis. So these are the epidermal cells. Here you can see the epidermis. Okay, these are the epidermal cells. This kind of cells you can see epidermal cells. And epidermal cells, they're single layer of cells. Single layer of cells. Okay. Generally, epidermal cells will be a single layer of cells. But those plants which are living in a very dry habit, like the areas, like some dry areas will be there, no, there. If these plants, some plants are there, means in that and all you can see thick, or the epidermis may be thicker. Okay, there you can see thick epidermis in order to avoid or protecting against the water loss. For that, epidermal cells will be more number of layers. So the entire surface of the plants, see the whole plant body that is having epidermis or epidermal cells. Entire plant body that is having epidermal cells, right? So this epidermis is protecting all the plant part. So epidermal cells on the aerial part, epidermis, that is the outermost layer. Okay, outermost layer of cells, which is called as epidermal cells. Okay, and in dry conditions plants are there, means usually it is single layer of cells. And in desert and all, desert type of plants and all, there you can see multi-layer epidermis also, and that is protecting entire surface, entire plant surface. Okay, so the aerial parts of the plant, often it will secrete a waxy water resistant layer on the outer surface of this epidermis. Okay, so that is giving protection towards the loss of water. So outer to that, the epidermis cells, they will secrete a waxy substance to their outer surface. So that is giving protection against water loss, then the mechanical injury, then other microorganisms which are going inside. So from all this, it is giving protection. Okay. So because of all this property, the epidermal cells, they won't be having any gap. They'll be having continuous cells. See, here also you can see continuous cells without giving any space or intercellular space or gap. So most of the epidermal cells, here you can see epidermal cells are, most of them are flat. Generally, they have flat cells. Okay. And here we can observe small pores, right? Small pores, and these pores are present in the epidermis of leaf. On leaf epidermis, you can see more pores like this. Pores on epidermal surface of the leaves. So these pores are called as stomata. Okay, stomata, you can see here, you can see many stomata which is present on the epidermal cells. And these stomata, you can see two kidney shaped cells. Two cells which are kidney shaped or bean shaped structure you can see here. And this is for gaseous exchange. Gaseous exchange, that is the role of stomata. So these pores are called as stomata. Stomata is enclosed by two kidney shaped cells which is called as guard cells, and these are very important for the gaseous exchange. Okay, gaseous exchange with the atmosphere. And the transpiration that is also taking place with the help of this stomata. Okay, so these are stomata, kidney shaped or bean shaped cells which are present on the in between the epidermis cells on the surface, right? And in leaf epidermis it will be more, and these are helping for gaseous exchange. And these cells which are called these kidney shaped cells, they are called as guard cells, and two cells will be present, right? And this is helping for the gaseous exchange. Okay, so that's all about the simple permanent tissues. So parenchyma, collenchyma and sclerenchyma we have learned. Then we are learning about the epidermal cells also, which are single layered generally, and multi-layered you can see in some kind of plants which are present in desert and all. Okay, that is to prevent water loss. Okay, so that's about the simple permanent tissues. In the next class, we'll be learning about complex permanent tissues. Okay, thank you.